Hello and good afternoon friends, welcome to the CEC EduSec live lecture. Yesterday we talked about various discoveries made in the field of biotechnology, and we discussed about various techniques which were used since 1980s. Today also we are going to know more about all the inventions and discoveries in this very field, and we'll start from 1980s, that is mid 1980s till now. And for this we have again with us in our studios Dr. Amit Bhattacharya. He is assistant professor in Ramjas College, as well as he has worked on microarray SAY technique on cancer biology. For this, I would like to congratulate him, as well as one interesting thing today is going to, which is going to be on this very lecture, that he is going to show you something very interesting. But I'm not going to tell you right now what is that interesting thing that which he is going to show you. But before that, I would like to welcome him and would ask him, sir, please continue the lecture further from where we left yesterday.
Thank you, ma'am. Good afternoon, dear students. I will continue the lecture which I started yesterday. Yesterday we covered most of the techniques and the methods which were discovered till 1980s. Yesterday we discovered lots of techniques or the methods which were discovered and which helped us to go further beyond the post-genomic era. We discussed about the electrophoresis, which gave rise to the blotting techniques, the restriction enzyme which gave rise to the recombinant DNA technology, the chemical structure of antibody which was discovered, which helped to get us to the ELISA technique, the DNA structure which was deciphered by Watson and Crick and Wilkins, which helped us to go to the next level, which is the DNA sequencing. Today we will be discussing about more advanced technique and more relevant technique to the medical and the pharmaceutical world. And basically we will be covering about the areas which are the applicative biology or the genetic engineering which comes up into the play. The first major technique which was developed or invented in the year 1984 was DNA fingerprinting. We all are aware of this DNA fingerprinting, or we have heard about this DNA fingerprinting technique. This fingerprinting technique is also called as the DNA typing technique. This was discovered by the British geneticist Alec Jeffrey. What he used is, he used the concept that each of our chromosome contains certain repetitive sequences like AATC, and these repetitive sequences are present within or around the genes. So what happens is, these repetitive sequence number also varies from one person to another person. Let me show you a zoomed in image of a human chromosome. If you see this human chromosome, a particular segment of the gene part has been focused into. In this gene part, there are different restriction endonucleases cutting sites present. And in between two restriction endonucleases site, there are 4 VNTRs which are present, which are called as variable number tandem repeats. As the name suggests, it means that there are tandem repeats, means there are repetitive sequences like ATC like this, and the number varies between person to person. So over here, if you see the right hand side image, there are 3 people's DNA which has been taken, a zoomed in image has been taken, in which between 2 restriction sites, in the first person there are about 14 VNTRs present, in the second person there are about 9 VNTR present, and in the third person there are only 4 VNTR present. So all of us are having this VNTR within our chromosomes, but the number is varying. So what Alec Jeffrey used is, he used this concept that due to which the DNA fragment size will differ between all of us. So if we restrict digest each of these fragments, they will be electrophoresed at different level. Like we have already studied the gel electrophoresis, the smallest fragment which contains 4 VNTR segments will move at the bottom, while at the middle part the second person's VNTR will come, which is having the moderate amount of 9 VNTRs, and finally at the top will be the VNTR of the person who are having 14 VNTR. So all of us are having this VNTR, the only difference is the number of repetitive sequence or the segments or the red blocks in us. So this was which was the primary thing which helped us to go into the forensic science biology. We all have heard about the forensic science crime cases been solved with the help of the DNA fingerprinting. So this is the very concept which is being used in the forensic science sample collection. So suppose at one of the places where the crime has been made, over there we get the DNA of the culprit. If we see the DNA of the culprit, over here I have shown 3 pairs of homologous chromosomes, and if you see all the 3 chromosomes, the red, green and blue chromosome, there are different number or different segments of VNTRs which are present. So there is a variable number of VNTRs among the chromosomes also. And on the basis of the information gathered in and around, the police caught 3 people on the basis of suspicion. But it is not confirmed which of these persons have done this crime or the murder thing. So what the crime investigation people do is they take out the blood sample. From that blood sample, the DNA is taken up. From all the 3 individual persons, the blood samples are taken. It is restricted, digested, and if we see individual A, B and C, we can clearly see there is a difference in the number of VNTRs which is being shown in the white blocks in all the 3 persons. So what we are seeing over here is the VNTRs are differing in the numbers within the chromosomes as well as among the individuals. So all of us have a very different varied composition of VNTR in our genes or in the same chromosomes. So when we restrict, digest it and go for a gel electrophoresis, we run it in different lanes. Like the sample from individual A was run on the A well, B from B, individual B into the B well, C from the C well, and the F well we run the forensic science sample DNA. So if you can see over here, the B sample is having a similar kind of pattern which is found in the sample which is collected from the forensic side. So we can clearly say that the B is the individual who has made the crime or has done the crime, and on the basis of this thing we can arrest that person, and now we have got enough evidence because the DNA evidence is regarded as one of the most trustable evidence, and the person B can be arrested for this making this crime. The forensic science, apart from forensic science, this DNA fingerprinting is also now being used for checking the paternity and the maternity fights also. So the DNA fingerprinting has enlarged its niche into different fields, and it is being now actively used in the different fields. The next major invention came up in the year 1987, for which Kary Mullis was awarded the Nobel Prize in Chemistry in 1993. He discovered the polymerase chain reaction. The main characteristic feature of this polymerase chain reaction is if we have got a very small amount or sample of DNA, we can amplify or make a billion number of copies of the DNA within 1 or 2 hours. So within 1 or 2 hours in a test tube or a append-off, some small test append-off, we can make billion copies of DNA from a single strand or single double-stranded DNA. Now, how exactly this works? This works exactly in the similar way like we discussed in the DNA replication yesterday. Over here, if you see, there is a DNA parental strand which is there, and there is a primer which is attached. And one after another, with the help of the DNA polymerase, the nucleotides are coming and forming a phosphodiester bond with the earlier attached nucleotide, and it is forming the next daughter strand, and the DNA double helix is being formed. So this is the case where the DNA replication is being done. So DNA replication, what we require for DNA replication is a DNA template strand, which is a single stranded DNA strand, onto which the next strand or the complementary strand will be formed, the primers which will be the forward and reverse primers, DNA polymerases which helps in the replication process or the amplification process, and the nucleotides which will be the deoxy adenosine triphosphate, deoxy cytosine triphosphate, deoxy guanine triphosphate, deoxy thymidine triphosphate. So this is the very basic concept of the DNA replication. Now, what Kary Mullis did is he exactly replicated this phenomena in a test tube or a small append-off. So what he did is he followed a polymerase chain reaction in a ependoff or a small vial, in which what he did is he followed 3 steps. At the first step, he went for a denaturation, means we have got the DNA of our interest present, so we need to replicate or form billion copies of this particular DNA segment, so it has to be denatured so that it gets into single-stranded DNA. So at the first step of denaturation, which takes place at 94 degrees Celsius, the hydrogen bond which is present between the base complementary, they gets breaked up and both these strands get separated from each other. This takes about 1 minute. The second step which follows is the annealing, where the primers goes and attaches to these single-stranded DNA, and these primers goes and attaches to the region which are nearby to the region which has to be amplified. This takes place at a temperature of 54 degrees Celsius and have to remain for just 45 seconds. Over here we require the forward and reverse primer. They are named so because they will do the front and the reverse replication. That's why they are named as forward and reverse primers. And in the third step, we go for the extension or elongation, where the DNA replication process will start taking place. So the polymerase will come and attach to the primer, and the replication process will start taking place with the help or the additions of the nucleotides one after another, depending upon their base pairing rule, means the G will base pair with C, T will base pair with A. So let me show you this process in a better animation. So over here, the 3 processes which we discussed is the denaturation, annealing and extension. If we see the animation over here, the first denaturation is occurring, so at high temperature both these strands get separated out. Second, the annealing is being done where the primer gets attached, and in the final step the extension takes place where the DNA polymerase goes and binds to those primer, and one after another the nucleotides get attached and move from one end to the other end. So in this DNA polymerase chain reaction, from 2 strands, we are at the first cycle we are going for 4 strands. So we are from 2 strands we are synthesizing 4 strands. So it is an exponential way it is increasing. So from 2 strands it's 4, from 4 it will be 8, 8 to 16, like this way it is increased. And it has been done that the polymerase chain reaction, if we run it for about 30 to 40 cycles, which takes about 1.5 hours, we produce a billion copy of that single segment of DNA which is of our interest. So this polymerase chain reaction has revolutionized the fundamentals of forensic science and all the investigation, because many a times at the forensic science forensic crimes sites, we find only a hair follicle, only a hair. So from that follicle we can extract the DNA, and that DNA can be amplified with the help of the polymerase chain reaction, and then subsequently can be used for the DNA fingerprinting and other processes. So this was one of the major discoveries of biotechnology. The next major discovery which came in way back 1991 was the discovery of the recombinant vaccines for hepatitis B. The hepatitis B viruses have been discovered way back in 1976 by Birnberg and Gatzuzek in the 1976, for which they were awarded the Nobel Prize. The structure of hepatitis B is similar like any of the viruses or the HIV viruses. The only difference is the hepatitis B virus is a non-retroviral virus. So the hepatitis B virus is the virus which infects the liver cells. So the hepatic cells are infected by this hepatitis B virus. The hepatitis virus have got different strains starting from A, B, C, D, E, but A and B are one of the most lethal strains which have been deciphered. So the hepatitis B virus has got a structure similar like the virus with a DNA double-stranded, partially formed double-stranded DNA present in the center. Then there are protein coats and an outer covering. On the top of the outer covering, there are surface antigens which are produced. present. So if we can see the electron micrograph of the hepatitis B virus, these hepatitis B virus are mostly infecting the liver cells. What happens is during the liver infection or these hepatitis virus, when they infect the liver cells, they become inflamed. And during this inflammation process, the liver cirrhosis also occurs. And it has been found that in 70 or the 80% of the cases where this hepatitis B virus infection goes for a prolonged period of time or their chronic diseases, the person has got about 80% of chances to develop liver cancer also. So these viruses are one of the most lethal viruses which occurs, and every year there are about 1 million deaths which occur due to this hepatitis B virus. Now, the vaccine against this hepatitis B virus was discovered using the recombinant DNA technology which we discussed last time. Now, before going into the recombinant DNA vaccine, let's start how exactly a vaccine functions when we inject it inside our body. These vaccines are more or less made through the surface antigen proteins which are present on the surface of the viruses. So we take out that virus protein and make it heat attenuated or non-infectious, or we can take out the gene and go for recombination. After this heat and attenuation or non-infection state, these viruses antigens, surface antigens, are then formed into the vaccine forms. These vaccines, when injected into the patient with the help of the syringe intravenously, goes into the blood circulation. Our blood circulation have got many of the lymphocytes circulating within the blood circulation, which are the part of the immune system or the immune response. So immediately these foreign surface antigens, hepatitis B surface antigens, are recognized by the macrophages, which engulf, present and process these things to elicit the second level of immune system, which compromises of the B lymphocytes. So the B lymphocytes are activated. If you see the blue color cell, which are memory B lymphocytes, they are activated by the presence of these virus coated surface antigens inside our body, and these B lymphocytes start secreting antibodies on the surface of it, and these antibodies are released into the blood. So now our blood supply has got a continuous supply of these antibodies present within them. So whenever we get any infection from this particular virus, these antibodies which are circulating within our blood will go and attach to these viruses, which are the foreign particles, and immediately go for the immune system without waiting for the next level of reaction, and the viruses will be invaded out from our body. So this is how the vaccine works. For all the vaccine generally works with the similar kind of mechanism. Now, the first recombinant vaccine for the hepatitis B was named as RECOMBIVAX HB, which means recombinant vaccine hepatitis B. This vaccine is a non-infectious vaccine because we have to make sure that the protein antigen which we are injecting within the body of the person or the patient should be non-infectious. It should not cause that disease, else the vaccine won't be of any use. So the basic structure of the hepatitis B virus is it contains a DNA, and if you see on the surface, the outer surface of the coating, there are red color surface antigens which are produced. So some part of the DNA codes for these surface antigen. These surface antigens are very important for these viruses because this is through these surface antigens these viruses get into the human cells. Like for example, the surface antigens present in the HIV viruses helps us this virus to get into the CD4 lymphocyte cells. So these surface antigens plays a very important role in the immunogenicity or pathogenicity of these viruses. So what the company did is, the company which manufactured this first recombinant vaccine was the Merck. So what this company did was it isolated the gene which coded for this hepatitis B surface antigen. So this DNA was isolated, and then this was put into a plasmid, which was a cloning vector isolated from the bacterial system, and a recombinant DNA construct was prepared, just like the recombinant DNA technology which we discussed yesterday. The recombinant DNA construct was then transferred into the yeast cell, which are the eukaryotic cell lines. This transformation takes place through various transformation techniques. We used over here the yeast cells because this is a eukaryotic cell line. So the proteins which will be produced from this particular foreign DNA or the recombinant DNA will be of eukaryotic type, containing all the post-translation modification, because in the eukaryotes there are lots of post-translation modification in the protein occurs which help in the proper folding and proper structural arrangement of these proteins. So these recombinant yeast cells which contains the recombinant DNA construct were then put into the fermentase, in which under proper environment or the growth medium factors, these cells started growing or dividing. And subsequently what happened that these recombinant DNA construct used the yeast cell mechanism to form the hepatitis B antigen within these yeast cells. So these hepatitis B antigens were taken out or lysed out from the cell, purified. Subsequently they were checked whether they are non-infectious or not, and when the scientists found that they are non-infectious to the human injection, then they were packaged into bottle and manufactured to the market. And these are the vaccines which are now being used intramuscularly for treating or for diagnosing the hepatitis B viruses. So this has become one of the revolutionary thing in development of vaccines, the recombinant DNA technology. The next major discovery which took the world around was the discovery of the microarray. What the microarray means is, micro means small and the array means sequence, one after other. So what the microarray did is, in a small chip or a small microscopic slide, all the genes from our body were encoded in this particular gene, in this particular slide. So the microarray helped to go us into the next level of the genomics, in which all the genes from our body, the whole genome of our body, was then transferred onto a small chip. Let me show you how exactly a microarray is. A microarray has a chip in which it looks like something like the microprocessor. Over here in this microarray contains lots of grids. There are a number of grids which are present in these microarray. So each of these grids have got lots of spots present into it. And each of these spots, if we further focus down into each of these spots represents one gene. So if you can see the topmost picture of the microarray, there are 4 spots present, and each of these spots are representing one gene. So each of these are having the DNA probe for the that particular gene. So this is the next level of genomics which came into the picture.
Yes, sir. As you are explaining about this particular chip, if we could have a demonstration of it, it would be very beneficial for our students, the viewers who might be watching us.
Yeah.
So could we get the demonstration of this
surely.
particular chip?
So I have got a microscopic microarray chip which is present with me. If you can see, this microarray chip has got a grids which looks something like the microprocessors, and each of these microarray chip, there are about 30,000 spots which are present, and in each of these spots they have got the DNA probes present into it, and these DNA probes are the single stranded DNA probes which represent a gene. So in total there are about 20 to 30,000 small spots which are present, and into each of these spots there are genes probes which are present. Each of these spots carries about 10,000 clones of one DNA probe. So you can just imagine how the science have got revolutionized in case of miniaturization. Means all the genes from our body can be now loaded onto a small microscopic slide, and we can use this microscopic slide for checking the disease conditions, whether the patient is going to suffer from cancer or whether what kind of cancer the patient will suffer, or which stage of cancer the patient is suffering from, or which are the diseases the patient is suffering from, everything. We can get a comparative and wholesome high throughput imaging of all the genes at a single go. So this is really an amazing technique which was developed by Max Clara.
This is all about this particular chip, and it can detect any kind of disease, or particularly for the cancer?
No, it doesn't detect the disease. Actually, it tells us the level of gene expression in each gene. So we can easily say, suppose there are some genes which are necessary for some diseases, like a gene which codes for the human growth factor. If that particular gene, we can see in this microarray chip that this particular gene is repressed in that particular individual, so we can easily say that this person is suffering from lack of human growth factor. So with this help of this gene, means we can give a complete overview of the complete gene expression and what are the genes in total which will be upregulated, which are downregulated, or which are normally present in the normal condition and a disease condition. So we can analyze in total all the things.
As we discussed yesterday also, that in case of cancer, in the case of diseases like cancer, would it be possible to know in well advance that person is likely to suffer from this disease? Does it helpful in that manner?
Yeah, surely. I will come into some of the slides after this one, in which I will show you how in the case of the cancer, it goes and detects, and it helps us to say that which are the genes which will be upregulated, or in the cancer patient, which are the genes which are upregulated, and then we can subsequently say that if this gene is upregulated, in future this will go and regulate this particular gene. So at that particular time, we can start diagnosing the patient with the initial level of the drugs or the therapy. So this is a remarkable technology which is a helpful thing in the pharmaceutical industries also.
With the help of this chip, our students might be benefited, and they got the live presentation of such kind of technique which has been made in the field of biotechnology. Yes, sir, moving forward towards the lecture.
So let me show you how exactly a microarray works. Like suppose we have got a DNA sample with us, which is of a patient whom we want to know that which are the genes which are actively expressed in that patient. So what we do is, this DNA sample, we fluorescently label this DNA sample. And as I showed you, in the microarray, there are about 30,000 spots, and each of these spots represents one gene. And in each of these spots, there are probe DNA which are attached over there. This probe DNA is a part of the gene which codes for that particular protein from that particular gene. So we incubate this microarray chip with this sample DNA, and subsequently after hybridization and washing steps, we analyze it under the microarray processor and the imager. So what we get is an image or the fluorescence imaging of that particular gene, and we get a spot at that particular spot. With the help of that particular spot, we easily can tell that this is the gene which is actively present in the sample of the patient, while the other genes are absent, and we can go to the next level of therapy, or the doctor can prescribe the changes in drugs, or he or she can tell you what are the other diseases the patient is likely to have, or what are the precautions the patient should take in future. Let me show you one of the example how it is being used in cancer biology. In a cancer biology field, like we have got a sample of a patient, and we want to know that which are the genes which are upregulated or expressed in a high level condition, or which are the genes which are downregulated or suppressed in the cancer patient. So we get this sample from the cancer patient and the samples from the normal healthy volunteers. We take out the RNA from the cancer patient, and from this RNA we make the cDNA, which is the complementary DNA, with the help of the enzyme which is called as reverse transcriptase. As the name suggests, we are making cDNA from mRNA, so the transcription process is reversed. That's why it is called as reverse transcriptase. So during this reverse transcriptase process, we add the fluorescent labeled nucleotides. So all the probes or the single-stranded DNA which will be formed in the cancer cells sample as well as in the normal cell samples will be fluorescently labeled, and to distinguish between them we label them in different colors. Means one, the cancer cells are labeled with red fluorescence molecules, while the normal cells are labeled with green fluorescence. And then we hybridize these fluorescence samples onto the microarray chip. If you have seen over here in the microarray chip, there were very small portions in which the samples can be loaded. So the samples are loaded at this very point, and the samples moves inside this microarray chip. They compete with the binding at these particular sites, and according to the level of expression of the gene between the cancer and the normal cells, the probe binding will take place. Now let us see how exactly the analysis goes about and what exactly we see in the analysis. When we focus down into one of the grid of this microarray, we can see in each of these spots different colors. If you see, some of these spots are red in color. This red in color shows that the genes, these particular genes, are upregulated in the cancer patient samples while they are normally expressed in the normal samples, which means that the mRNA which is taken up from the cancer patient contains a higher level of these particular genes level than the normal patients, and subsequently the probes numbers are much more higher than the probe number which is present in the normal cells. And that's why the red color is there, because red color, it is outcasting the green color, and we are getting a red fluorescence image at that particular position. Subsequently, if we get a green color, that shows that the number of probes which are present are higher in the case of normal cells than the cancerous cells, means these particular genes are down-regulated in the cancerous cells. And if we get a yellow color, which is a mixture of both the colors, it shows that this gene is normally expressed in both cancerous as well as in the normal state. So from these results, we get a complete imaging of the entire genome phenomena. So from a single microscopic slide analysis, we get that the person will be is suffering from which kind of diseases or which kind of genes are upregulated or downregulated, and we get a complete overview of all the genes which are expressed or up-regulated, down-regulated, or normally expressed like a normal cells. So this microarray has certainly revolutionized the next level, where a complete or whole genome of our body, which contains about 20 to 30,000 genes, have been put into a small microscopic slide in which we can detect all the genes at a single time. The next major discovery came up in the year 1997, which was called as fluorescence in situ hybridization. As the name suggests, fluorescence tags are being attached. In situ means in position or at that particular point, and hybridization is we are using some probe to hybridize to that particular point. In this method, instead of taking out the DNA, RNA or protein from the samples, we take up this cell as a whole, and we can analyze this cell as a whole and its chromosome within a on a slide. So what the scientists did is they can now find out the chromosomal abnormalities present on these chromosomes, because on a single slide we can take out all the chromosomes from this cell, and then we can stain these chromosomes with different probes, and we can easily find out which are the genes which are duplicated or which are the genes which are deleted. So this has been called as the fishing out of the genes which are present in the chromosomes or which are not present in the chromosomes. Now let me show you how exactly we do the fluorescence in situ hybridization. We take a sample from the patient. the sample cell is taken onto a slide, it is fixed onto a slide, then the cell membrane and the nuclear membrane are lysed out so that all the chromosomes are taken out from the cell and they get arranged onto the microscopic slide. Then we go for the denaturation process, in which the double-stranded DNA present in the chromosomes are converted to single-stranded by cleaving the hydrogen bond between them. Then we hybridize these chromosomes with the probe which are specific for specific gene of our interest. These probes are single-stranded, so they will go and bind to the complementary region wherever the gene is present, and these probes have got a ligand present on the top of it. As the hybridization process is over, we put these slide along with the antibodies, which is labeled with a fluorescent dye on the top of it, and we go for a fluorescence imaging. So on the fluorescence imaging, we can see the chromosome point at which the probe has gone and attached to it. So by this process, by looking at the fluorescence microscope image, if there are 2-3 spots, it shows that the gene has been translocated to other areas or it has been duplicated. And if we see that there is no chromosomal probe spot present in the imaging, it clearly tells that the gene has been deleted from our sample. So we can easily tell that which kind of deletion the patient is suffering from, because there are lots of diseases which happen because of the deletions, transversion, translocations or mutations. So we can all take out in the chromosome level. So let me take you to one of the example of this fluorescence in situ hybridization. In this example, we have taken an example of cancer pathology. Now most of the cancer biopsy which are being done are using this fluorescence in situ hybridization. Whenever the doctor wants to say that whether the patient sample is having any kind of cancerous growth or not, the sample is sent to the laboratory where the cancer biopsy is being done. So this is being done in a similar fashion. The cancer tissue cells are taken onto the slide. These cells are lysed up so that the chromosomes comes out on the slide. Then the chromosomes are denatured with the help of the denaturing agents and then incubated with the probe, and the probes are finally visualized under the fluorescence microscopy whether the probe's colorations or the fluorescence is present or not. Over here I have taken an example of advanced prostate cancer. If you can see at the picture A, there is a chromosome which is present. In this chromosome there are certain part of gene which codes for a gene which is called as tumor suppressor gene. These tumor suppressor genes are very essential genes for suppressing the tumor growth within the body. So whenever the patient suffers from any tumor, these are the genes which are deleted from the chromosomes, because until unless they are not deleted or inactivated, the cell growth won't occur. So over here, if you see, there is a chromosome tumor suppressor gene which is present on the adjacent side of the part. There are certain flanking regions also present. To these flanking regions, there are probes which has been made, which have been fluorescently labeled by different color, like green for the probe A and blue for the probe B, while the tumor suppressor gene has been labeled with the probe red in color. So what we do is, after we prepare this slide, we denature the chromosomes, we incubate this slide with these probes. So these probes will go and bind to the gene of our interest or the specific gene where the complementary sequences are present. So in a normal condition, if we see in normal condition, as we have got a pair of genes, so there will be two set of these probe images which will be coming out. So in one chromosome we can see the red, green and blue image, and in other chromosome we can see another set of red, green and blue, which shows that the genes are intact and the probe have rightly bound to those particular genes in the chromosome. Now in the second case, if you see, there is a hemizygous loss, which means in one of the chromosome of the pair, the tumor suppressor gene has been lost. So there is no addition or no binding or hybridization of the red color probe. So we cannot see any red color fluorescence imaging in the second set of the image. So, and finally, there is a homozygous loss where both the set of the tumor suppressor genes are lost from both the chromosomes. So, in the image, if you see from both the chromosomes, the red color fluorescence is not there. So, this clearly shows that when a normal gene is present, there are presence of 3 set of the probes which are intact. When the patient moves to a hemizygous loss, which means that the patient is going to the next, is starting with the cancer process, means the gene has got disrupted in one of the chromosomes, so it goes into a hemizygous loss. And finally, when the cancer moves to an advanced state, both the genes are lost from the chromosome pair, so we cannot see any fluorescence imaging in any of the chromosomes. So we can clearly say that the patient have moved to the advanced level. Now, this is being shown a picture over here where we can see in many of the fluorescence imaging cells, we can see that the complete set of red, green and blue has been deleted out, which shows that all the genes from one of the chromosome set have been deleted, which shows that the patient has almost moved to the next or the advanced level of the cancer state or the prostate cancer state. Now, the scientists have been very innovative in innovation the things in which they have subsequently painted the entire chromosome. So, all the 24 set of the chromosomes have been painted different color. So, the recombination DNA technology and these advanced technology are putting up the inventions and innovations in a completely different perspective. To end the presentation, I will just focus into 2 of the major projects which were completed in this 21st century. The first one came in as the Human Genome Project, in which whole of the human genome or all the chromosomes genes were decoded and it was completely sequenced. This project started way back in 1990 and got completed in almost in the year 2005. It took 15 years and all the chromosomes have been sequenced out. And if you can see over here, there are about thousands and thousands of workers which worked day and night. And in one of the picture, if you see, this is the laboratory where the DNA sequencing occurred. There are about 1,000 DNA sequencer which were placed one after another, and the robotic arms were used in which the DNA was taken out from the bacterial cultures after they were processed or amplified. Then they were cleaned, sequenced. So the robotic arms and about 27 international collaborators worked on this field day and night. The next major discovery which came recently was in 29th May 2014, where the complete human genome protein sequencing has been done, and to a pride we can say that we can see lots of Indian names in this paper. This has been published in Nature 2014, and about 7 of the Indian universities and institutes actively participated in deciphering all the sequence of the complete human genome thing. So they have taken samples from the adult, all the tissues from the sample, all the tissues from the adult, right from heart, kidney, liver, lungs, from the fetal tissues also they have taken the samples. They have taken samples or the proteins from hematopoietic stem cells which are actively mutated in the blood cancers, and the basic workflow which they followed is the protein was extracted from each of these organ tissues. They were SDS paged, which we discussed last time, which was the sodium dodecyl polyacromide gel electrophoresis. Then they went for the digestion with the help of the trypsin digestion. The polypeptide chain was digested into small chains. Then they went for the fractionation with the help of the RPLC, which is reversed phase liquid chromatography, and finally the sequencing was done with the help of tandem MS. So in total what they have found is they have found the proteins which are coded from about 17,000 genes. So 84% of the gene proteins have been coded, and till date this is one of the largest coverage of the human proteome reported so far. So these 2 projects, along with all the techniques, microarray, DNA sequencing, DNA fingerprinting, southern blotting, PCR has certainly revolutionized and opened the field into a next level of biotechnology and genetic engineering, in which the scientists will be working and decoding these analysis and making up the drugs, pharmaceuticals, things, and transgenic animals, transgenic plants. And what we require at this point is the bright and the intelligent students like you all into the field of science, who can take this venture into the next level and into the next field of revolution. Thank you very much.
Definitely, we pray and we hope most of the students would be taking biotechnology as a career. But if we are talking about, or we are talking about this particular thing, career, as we were talking before that, how earlier it was a challenge to get into a CPMT exam and become a doctor. How do you feel, and what you feel there is what kind of difference between a biotechnologist and a doctor?
Okay, so the first and the foremost thing is, there is no need to be getting demoralized if you don't get or clear down the CPMT, DPMT or whatever medical entrance exams, because the medical doctors are the one who prescribes or read about these drugs and then they prescribe these drugs. But a biotechnologist is a level ahead of these doctors, because the biotechnologist is the one who actually makes and reads about these vaccines or the drug. So he is the one who discovers and invents the drug. So until unless we don't have any biotechnologists who will be inventing or decoding these drugs or the vaccines or various things, the doctors won't be able to prescribe the next level of drugs to the patient. So at any point of time, the biotechnology is certainly a next level of these medicos thing. And to a surprise, you all will be knowing that now most of the doctors who have done their MBBS degrees or the MD are now getting into the biotechnology field, where they are working on the development of these vaccines, drugs in the major pharmaceutical companies. So they can use those expertise of MBBS as well as the science. But a normal graduate student who does his normal graduation in PO science, post graduation, and did do his PhD is also a very good boom to the biotechnology field, because he or she knows about what exactly the mechanisms occurs, how exactly the things can be manipulated in the genes, because he or she has got a in-depth knowledge about all the techniques and everything. So he can apply these things to the next level and can revolutionize the biotechnology field.
Definitely, if you have it in you that you have to discover something new, definitely you can go for the biotechnology field. But as far as if I talk about you, that you have worked on the microarray HB technique, I would like to ask you, as you have shown this chip also, what is the life of this chip?
This chip actually are being packaged or shipped into a very sealed consignment. So they have to be kept in a completely sterile environment, and they generally can be kept for 1 or 2 months. But we cannot use them after that, because there will be a chances of getting these chips contaminated. So if we contaminate these chips, we won't be getting the results which will be the actual results, and these contamination won't help us to confidently say that these are the things which are coming rightly or not. And these chips are now customly available in the market. So you just go to the site of the companies who produces these chips like Illumina, Affymetrix. You go to their sites, you just fill in your query that what kind of genes, or you want all the human genome in this chip, or you want certain particular genes or a sequence of genes into these chips. They will custom made to you and package and ship it to you. It generally takes some time for making this chip, but you can subsequently ask them the fresh chips and you can use them into your experiments. But the chip generally are used whenever the shipping arrives. They have to be used within few months time, because the sterile environment and the sterile conditions is a major factor, because they might get as a confounding factor or a troubling factor in the subsequent analysis of these microarray data.
And as far as the cost of the chip is concerned, as you have told that there are a number of and numerous companies who produces such type of chips, does the cost of the particular chip varies from one to another?
Yeah, the chips are generally, one of the disadvantage of this microarray technique is the high cost. These chips generally cost from 50, 70, 80,000 lakhs, 1 lakh, 1.5 lakh, something like this. So these chips generally cost about lakh to us. So this price factor is a major concern, but the companies are trying to minimize this factor by decreasing the price values. And depending upon the structure, what we want in the genes which we want onto the chip, the price values may fluctuate, because if we custom made the chip to a very specific level or very specific kind, the prices may increase, because then we are telling the company that these are the particular genes which you have to probe onto the top of the chip. So the company have to work on an extra level to put those particular genes onto it.
As you discussed and told us about this life of the chip, that yes, of course, there is a clear life for this particular chip. Do we have to carry some working condition for this chip while using it?
Yeah, all these biotechnology work are done in a sterile environment. Means most of the work which are being done in the biotechnology are done in a laminar hood or a sterile environment, so that there is no flow or the contamination of any microorganisms or any dust particles. Because if we go through all the techniques, when you will be starting working in any of these techniques, you will be seeing that if you have any kind of minute dust or contaminants present within your sample, your whole experiment get washed away. And each of these experiments takes about 1.5 2 months to carry out, and before carrying out this experiment, it takes about 5 to 6 months to standardize the process. Means which are the proper conditions which has to be put, like what are the proper amount of chemicals which has to be used, is this chemical acting a little bit more or is it less. So all these things have to be standardized. So when you start with the experiment, you have to make sure that all these things are done in a proper environment or a sterile environment or a sterile hood or condition, so that there is no confounding factor of any impurity or contaminations coming into play.
Now, apart from this chip, and when we are discussing about all the discoveries and innovations made in the recent past, or we are making certain discoveries, do you feel this biometric machines which are usually used in the offices is a part of a biotechnology?
No, actually this is not a part of the biotechnology. This uses the fingerprints of the person, means the fingerprints which are present in the thumb. So this doesn't use the biotechnology, so it should not be confused with the DNA fingerprinting. They are the fingerprints which are being present in our normal fingerprints, but subsequently the name has come just because we use similar kind of thing, because all of us differ in the fingerprints which are present over here. So that's why, just to have a synchrony of the name, it has been named fingerprinting. So that should not be confused. Like in the northern, southern, western blotting, only the southern blotting was discovered by the scientists called as southern, while the northern and the western were not discovered by the scientists who were named northern or western. They were discovered by Alvine and Brunet. So they should not be confused. This is just to make a correlation or a synchrony with the name. So they use these names.
But if you talk about the Department of Pathology, the way various machines and various techniques have been developed, can we say that yes, pathology as a department as a whole, or the discoveries made under it comes under the biotechnology?
The pathology department actually covers up lots of things. So along with the biotechnology, there are studies of cytogenetics which also come into play. So as I showed you the fluorescence in situ hybridization, now the pathologist labs are majorly checking these biopsy of the cancerous growth or the cells with the help of these biotechnology technique. So along with the microscopic staining technique, they are confirming it with the help of the biotechnology technique, so that because whenever we say to a patient that he or she is suffering from a cancer, we have to be absolutely sure whether the patient is suffering from that particular cancer or which stage of the cancer is present. So the pathologist department, along with the normal staining or the cytogenetic study, they are taking the biotechnology techniques also, so that everything is confirmed before starting any drugs, because the chemotherapies are very lethal to the normal cells also. So we have to be absolutely sure before starting any chemotherapy drugs for the patients.
Definitely, definitely. And now we have, I discovered certain terms which are the biotechnological terms, and most of the students who are watching us may not be or may be aware with the certain terms. Just for their revival of the thoughts, their revival of the concept, I would like you to please briefly explain the terms such as DNA replication, lymphocytes. Next is eukaryotic cells. What are these concepts? If you could give us a brief insight into it.
Okay, fine. So the DNA replication is one of the basic process in which, if we have a single strand of DNA, these complementary strand of the DNA can be produced, and this process is called as replication. Means from one parental strand we are synthesizing or making the other daughter strand. So this very basic process is called as DNA replication, which occurs both in prokaryotes and eukaryote cell. Prokaryotes means, as the name suggests, pro means primitive, karyotes means the cell. So the primitive cells like the bacteria are called as the prokaryotes, while eukaryotes means the cells which have got a true nucleus. Eu means true and karyote means the cell nucleus. So the cells like the yeast cells, human cells, these all comes under the eukaryotic cells. So the prokaryotes and eukaryotes are defined by the compartmentalization of the cell organelles. And the other question you asked, ma'am, was the lymphocytes. Actually, the lymphocytes are the cells which actively play a major role in the immune system. So whenever in our body, if there is any cut or any infection which occurs, so these lymphocytes are continuously circulating in our blood circulation. So in our blood vessel circulation, along with the RBCs and the other factors, the lymphocytes are also circulating. So what this lymphocyte cells does is, it immediately recognizes these foreign particles and engulf within them. Means it digests these foreign particles within them, so that it can be removed out from the body and it doesn't harm to any of our normal processes. So these lymphocytes are one of the major players in the immune system, and they are categorized into B lymphocytes or T lymphocytes depending upon the specificity, the role and everything.
Because we have very less time left also, but in brief we can discuss about what are the future prospects in the field of this biotechnology. What do you think, what innovations could be made or can be discovered?
Means the biotechnology field has now got immense number of opportunities and things. So a student who wants to study the biotechnology can go into diverse number of fields, starting from genetics, immunology, biochemistry, because the biotechnology field, as the name suggests, is an interdisciplinary subject. So now it has become an interdisciplinary subject. Even students from physics background are also coming into biotechnology, are helping the biologists to develop the various machines. Like for example, the machine which scan the body for the cancerous growth, the PET, MRI are being developed by the biophysicist or the student or the scientists who have actively worked in the physics background also. So the biotechnology field is a global field which can be taken up by the students, and now the number of universities or the institutes have also increased in India. So the students can go from any kind of studies in biotechnology, may it be marine biotechnology or cytogenetics or genetic engineering, forensic
Definitely,
sciences.
we pray for the best and attempt for the best. And with this note, I would like to thank Dr. Amit Bhattacharya for giving deep insight into this very topic, and hope in future lectures also we will try to learn something new under this subject, that is biotechnology. Thank you, sir. Thank you very much for being with us.
Thank you very much.